Okay, good morning. We're going to go ahead and get started here. We've got a pretty fair number in already. So we're going to continue our discussion on chemistry today. And we're getting closer and closer to wrapping this up. We may wrap it up on Tuesday of next week. Just depends. I'm not going to rush it to see, just to get it done, But I'm hoping definitely by next Thursday, we'll be finished with the chemistry chapter. And I'm sure we'll all be glad to see it go. The lab this afternoon is going to be on the bones of the skull. We're going to take a look at this. Now, we're going to go through the detailed structure of the skull, bone by bone. And I'm going to talk a little bit about how, what the bone test that will come up next month is going to entail for you. Also, the lab test should have opened up today, and it's open for the next 10 days. It ends on the 19th. So I'll talk more about that when we get into lab too. Anyone have any questions on anything? So I assume you're all getting my emails with the links for the videos. I'm hoping that they work. So I haven't heard any complaints. I guess they must be if you're using them. So, and it's I'm happy to do that. It turned out to be a lot easier task than I thought it would be. So it's probably should have been doing it that way all along. So anyway, let me open up the screen and go into chemistry. Okay, so where we left off the other day, we were talking about chemical reactions and what causes the chemical reaction to work and things like that. And chemical reactions work simply when two or more reactants randomly run into each other. That's how a chemical reaction works. It's just molecule A runs into molecule B, and then they do something to each other, and then they end up with a product. They grab electrons, or they give up electrons, or they share electrons, or they kick out water, or they gain water. It's all random. And what brings those molecules together, they randomly occur. Now, some reactions give off heat. Some reactions are what we call exergonic. They're going to give off heat. They're going to warm up as they go. They're going to end up with less energy they started with when the reaction's over. When we burn a log, or we burn charcoal, or we burn gasoline, that's a chemical reaction. In this case, we control it because we gasoline in our car, or a log in the fire pit, or charcoal in the grill, or propane in the grill. Those are controlled chemical reactions. And they work because you're bringing you are bringing oxygen and propane together with ignition source in your grill, and the reaction is breaking the chemical bonds of the propane to release heat and light, and giving off carbon dioxide and water. It works because we have concentrated levels of propane present. But if you were to stand 20 feet away from your grill and open up the valve in the tank, just release the propane into the air, that wouldn't work, because the propane is so randomly scattered throughout it, just like it is in regular atmospheric air. Chemical reactions work because molecules run into each other. And when we have a reaction, we can give off heat, or we can require heat. If we burn a log or burn charcoal, we're going to break down molecules of the reactants and break chemical bonds and release energy. If we have an endergonic reaction, exergonic gives off heat, endergonic requires energy. Endergonic reactions require energy to work. You can't burn propane in your grill unless you have an ignition source. You have to apply energy to it to get it to work. You can't bake a cake without heating up the batter in the oven. The oven causes a chemical change to occur in the batter. And endergonic reactions absorb energy. They use the heat energy to make the ingredients in the batter move faster and react with each other. And we totally give a complete change. Because when you take a cake mix, this goes back to mixtures and reaction and here. If you have a cake mix and you mix all the ingredients together and mix it up, you've got the batter, but you could still physically separate the ingredients of the batter out. I don't know why you'd want to, but you could. You had enough filters, you could filter out all the ingredients. But if you put them in the oven, in the pan, hopefully, and turn the oven on, another important point, you're going to cause the molecules to move faster in that batter, causing reactions, chemical reactions to occur as the molecules slam together because they're moving faster. And we get a complete chemical change in the makeup of the batter, and now it's cake. And it cannot be filtered like it could when it was the mixture. It's now a product, and it has stored energy in it. You can't find the eggs. You can't find the sugar. You can't find the flour. You can't find the oil. It has now all been chemically changed because they've been randomly running into each other. Because if a cake didn't require extra energy, you could just pour it into a pan and set it aside and let it go, and there you have your cake. But it doesn't work that way. In endergonic reactions, we have more potential energy than we started with. So we have more potential energy from our cake than we do with the ingredients of the mixture. Now, a chemist will tell you that chemical reactions are reversible. I know we got into this the other day. Chemical reactions are reversible because the reactants can become the products and the products can become the reactants. Well, that's true to some extent, theoretically, in a chemical laboratory. But if you can't go backwards, that's more of the reality in the human body. You can't go backwards because you've usually used up the products. Chemical reactions in our bodies make products that are used right away. So we don't have products that can go backwards, or we don't have the energy to go backwards and reverse the reaction. So they are reversible in the lab, but generally not reversible in our bodies. Now, 3 things that can affect the rate of chemical reactions. These don't work for us in the human body. First of all, temperature. If you increase the temperature of a chemical reaction, it will go faster. Cake batter slams into other cake batter molecules at a faster rate at a higher temperature. If you bake the cake at 400, it'll get done quicker than at 200, I guess. I don't know. I don't try that stuff. But 200 degrees is not a drastic increase in temperature as 400 degrees is. At 400 degrees, the molecules are moving faster. Of course, if they move too fast, you end up ruining the cake, but that's a whole other story. Temperature increases. A rise in temperature increases the rate of the chemical reaction occurring. The more energy you apply to a system, the faster the reaction occurs. If you were doing the baking soda and vinegar volcano, what if you used hot vinegar? What if you had heated the vinegar up before you mixed it into the baking soda? It would be a much more rapid reaction than just the usual foaming out you get when you mix baking soda and vinegar and a release of carbon dioxide and water and salt. Increase the temperature, increases the reaction. We can't do that in the body. We are stuck at 98.6 or 37 degrees Celsius. We cannot increase our body temperature. So any chemical reaction in our body has to operate at 37 degrees Celsius. Maybe a little higher, maybe a little bit lower. You know, when we have a low-grade fever, we increase the efficiency of chemical reactions in our body. It helps to break down the bacteria faster that's causing an infection. But generally, all chemical reactions in our bodies are designed to operate at 37 degrees Celsius. Concentration plays a big role in a chemical reaction. If some is good, more is better. That's true in a laboratory setting. If you increase the concentration of the reaction of the reactant, the rate will go faster. Add more vinegar, add more baking soda, add stronger vinegar, add stronger baking soda, more concentrated baking soda. If you've ever seen the experiment for generating elephant's toothpaste that involves hydrogen peroxide, the elephant's toothpaste, you have the safe home version that doesn't even use hydrogen peroxide. Well, it may use the 3% that you get in the brown bottle. But if you've ever seen the elephant's toothpaste that comes out of the big graduate cylinder, it shoots up in the air and foams and foams and foams. It gives off a lot of heat and smoke and everything. And you have to do it in a hood or do it outside. It's very dramatic, especially if you put food coloring in with the it's detergent and iodine and vinegar. I mean, not vinegar, hydrogen peroxide, and some food coloring to make it look pretty. If you try to do that with regular off-the-shelf 3% hydrogen peroxide, it may work, but it's not going to be pretty spectacular. But if you get the concentrated hydrogen peroxide, the 70% hydrogen peroxide, the stuff you have to buy through the chemical supply houses, stuff you just can't walk into Walmart. I'd like some of that 70% hydrogen peroxide. Not going to get it. That gives you a reaction that is spectacular. If you've ever seen it in person or seen it on YouTube, it shoots the foam up in the air. The concentration has increased. The greater the concentration, the faster the rate. Why? Because these molecules are moving around, in the reactant, and the more you have of them, they may not be moving they may be moving at room temperature, but you got a whole lot more of them. And a chemical reaction is simply based on two molecules randomly coming together. The odds are much greater if you have more of them. So you have a greater concentration. The more you have, the more concentrated they are, the more likely they're going to come together. But we don't get that benefit in our physiology. We are dealing with very tiny concentrations, very small amounts. We can't increase the concentration. We're dealing with extremely minute amounts here. And particle size. If some is good, more is better, but in particle size, smaller is better. Because the smaller the particle, the faster it's going to move around. And that's what we want to see happening in a chemical reaction. We want to see molecules moving around rapidly, running into each other. Again, our particles are already tiny in chemical reactions in the human body. That can't help us either. So we can't raise our body temperature. We can't do anything about the concentration, and we can't do anything about the particle size. What we can do is use a catalyst. A catalyst is a molecule, a chemical molecule that causes a reaction to occur at a faster rate. It's usually an enzyme. Enzymes are what we call biological catalysts. We can't change the temperature, we can't change the concentration, we can't change the size, but we can flood an area with millions of enzymes. And what the enzymes do is they grab reactants together and they grab them and bring them together and inside the enzyme and force the reaction to occur. So while we can't do anything about size or temperature or concentration, we can make enzymes. And so we can go out and grab these random reactants and slam them together inside the enzyme, and the reaction occurs. We can do that as much as we want, because the enzymes aren't harmed. The enzymes, they bring two reactants together and the reaction occurs, and then they let go of the reactants, and then they go do it again and again and again and again. And so that's how we can make reactions go fast in our bodies. We use this thing called a catalyst. Okay. So this brings us up into the biochemistry. Now, up to this point, we've been talking about what we call physical chemistry. Biochemistry deals with human anatomy and physiology. And the chemistry here in this biochemistry is either going to be inorganic or organic. I should have said organic or inorganic. That doesn't sound awkward. Anything involving inorganic chemistry deals with water and salts and acids and bases. There are no carbon atoms involved in any of these molecules. There's no carbon in water. There's no carbon in salt. Acids and bases don't usually have carbon in here. So the inorganic compounds, the inorganic chemicals do not have carbon. Everything else are the organic molecules. Everything in organic chemistry is based off of carbon. In fact, organic chemistry is often called carbon chemistry. This covers and includes the carbohydrates, the starches and sugars, and grains and cereals and potatoes and all this stuff. Carbohydrates, that's an organic compound made of carbon and hydrogen and oxygen. Fats are an organic compound, also made of carbon and hydrogen and oxygen, and occasionally phosphorus thrown in there too. Proteins, carbon, hydrogen, oxygen, and nitrogen, and the nucleic acids. Again, carbon and hydrogen and oxygen and nitrogen and phosphorus, all assembled in a very large molecular chains called the DNA or the RNA of the body. They're very large molecules. They're all covalently bonded. All these organic compounds, even the simplest of the organic compounds, are 4 or 5 carbon atoms long, 3, 4, or 5 carbon atoms long. So the organic compounds, have multiple shapes and sizes, all covalently bonded with the carbons, all involving hydrogen, carbon, hydrogen, oxygen, sometimes nitrogen and phosphorus, sometimes carbon, hydrogen, oxygen, and phosphorus. So the most common inorganic compound is water, H2O. 70% of our body volume as an adult is water. 80% when we're children, 60% when we become elderly. Older people, the elderly have lower body water content, which explains why their skin is wrinkled. Their water wrinkles because of a lack of what we call turgor, water filling up the spaces in here. And so their skin shrivels due to a lack of water. So their body water is about 60% as opposed to 70% of, for adults in our age, or 80% in children. Water is essential for us. Water has some very unique properties. It has what we call a high heat capacity, a high heat of vaporization. It is a polar solvent. It is reacting, and it is cushioning. A water molecule can bind with 4 other water molecules. Amazing property there. Let's take a look at these. The heat capacity. Water has the ability to absorb heat or release heat without a significant temperature change in the body. We can maintain 37 degrees Celsius in our body very easily on a hot day. We can absorb heat, we can release heat, no change. We maintain stability in our bodies thanks to this 70% water. It takes an awful lot of heat to change the temperature of water. We use water to cool us. We sweat on a hot day. We perspire on a hot day. And this is how we stay cool. Water, it takes a lot of heat energy to vaporize water. And so that's how we cool ourselves. Water evaporates off our skin. And that water evaporates off our skin thanks to the high heat. And we are dissipating the heat when water evaporates. That's why we perspire, why we sweat, to cool us down so that the water can evaporate and release the heat. Water functions as a solvent. It's a polar solvent. Water molecules have positive and negative ends. It can break apart salts very easily. The salts have positive and negative charges. And so we use water as the solvent in our plasma. Our plasma is the liquid portion of our blood. It contains the red cells, the white cells, and the platelets. It contains the hormones and the vitamins and the glucose, and sodium and potassium and other ions floating around in here. It is the universal solvent. It also protects large molecules like proteins, which is a water layer around it. And we also see that we dissolve air, oxygen, and carbon dioxide in water that go across our lungs to make it easier to move across our lungs. So water functions as this solvent and transport medium for us. Without water, our blood cells couldn't move because our blood cells float in the plasma. Now, salts. Salts are an interesting topic here. Salts are ionic compounds. Sodium chloride is an ionic compound. It consists of a sodium ion and a chlorine ion. The sodium ion has lost an electron. The chlorine ion has gained an electron. Sodium chloride is held together by this ionic or magnetic electrostatic attraction, if you will. If you drop a salt crystal into water, it will disassociate. It will separate into the sodium ions and the chlorine ions. It will separate into the positively charged cations and the negatively charged anions. And these conduct electrical currents in our bodies. We call these electrolytes. They allow our cells to have charges. Muscle cells have a charge on their cells of minus 90 millivolts. Nerve cells have a charge of minus 70 millivolts. Most of the sodium in our bodies is found outside of our cells, in the fluid surrounding the cells. And most of our potassium is found inside the cells. These are responsible for generating the charge on the membrane of these various cells. We call them electrolytes. These ions can conduct electricity, and we are electrical. The ions that we need the most of are sodium and potassium and calcium and iron. Iron we need because it's the center of the hemoglobin molecule. Hemoglobin carries oxygen. The oxygen atoms, oxygen molecules bind to the iron ion. And so each hemoglobin molecule has one iron in it, and it can hold 4 oxygens. Sodium, potassium, and calcium. Notice we leave off chlorine. Chlorine is not a major player in the electrolytes. It's there. It doesn't do a whole lot. The key players are going to be sodium on the outside of the cell, potassium on the inside of the cell, calcium outside the cell. In the heart, we use calcium in lieu of sodium. We'll get into that a lot in AP2. These ions are very, very important to us. We depend on these ions for nerve impulses, for muscle contractions. Gotta have them. We depend on sodium for regulating our blood pressure. Too much sodium raises our blood pressure. Too little sodium causes our blood pressure to drop. Sodium levels in our bodies tell us when we're thirsty and tell us when we have to pee. When we say dissociation, it means they break apart into their 2 separate ions. In this case, sodium chloride is attacked by the positive and negatively charged water molecules, and the positive end of a water molecule, the hydrogen end, will grab onto the negative chlorine and pull it apart from the sodium. The sodium will be grabbed by the negative oxygen on the water molecules. Remember, water molecules are polar. And that's why when you take sodium chloride and add it to a beaker of water, the sodium chloride, the salt disappears. It goes into solution instantly because the sodium chloride has a positive and negative charge. Holding it together, it separates out because it's attracted to the positive and negative ends of the water molecule. It goes into solution very easily. Sugar doesn't do that. Sugar is not positively or negatively charged. Sugar is not an ionic compound, it's a covalent compound. There is no positive or negative end on a sugar molecule. There's no place for the water to attack to pull the sugar apart. So it takes more effort to get sugar to go into solution than it does salt. To get sugar to dissolve, like if you're making sweet tea, and you want to dump in 5 pounds of sugar and 10 pounds of tea or whatever you're making, that tea has to be hot. The water molecules have to be moving faster and faster than room temperature to break apart to get the sugar to go into solution. You have to put energy into it. Otherwise, the sugar is going to collect in the bottom of the container or in the glass, who's dumping sugar in your glass. Ionic compounds go into solution very easily because they dissociate quickly thanks to the positive and negative charges on the water molecule. Another category of inorganic compounds are acids and bases. They're considered electrolytes because they have positive and negative charges. They will dissociate in water. Sodium hydroxide, a very powerful base, will separate in water, will go into solution as sodium ions and hydroxyl, the OH ions. Hydrochloric acid will dissociate into the hydrogen ion and into the chlorine ion. The hydrogen ion has a positive charge and the chlorine ion has a negative charge. Acids in our bodies include hydrochloric acid. We have it in our stomach. It dissolves proteins, or at least it starts the process of breaking and digesting protein in our stomach. And carbonic acid. carbon dioxide that's generated in the cells is transported to the lungs as carbonic acid. It combines with the water in the plasma to form carbonic acid. It travels to the lungs in that form and then gets released as CO2 at the lungs as the plasma and blood come in contact with the lung tissue. Carbonic acid, it's a very weak acid, but it's still an acid. And CO2 will form carbonic acid in the body. And that's also what tells us to breathe faster after exercise, because carbonic acid makes our blood more acidic. Any acid, with carbonic acid, when it forms, will make our blood more acidic. And our brain says, hey, our blood's more acidic. It's probably because of carbon dioxide. Let's breathe faster to blow off the CO2. So we breathe faster after exercise, not to get more oxygen, because we got tons of oxygen in our blood. And we have more oxygen than we need, because we ever only use about 20% every time the blood makes a full circuit through our bodies. We still have plenty of oxygen available. So we're not breathing fast to gain more oxygen. We're breathing fast to get rid of the CO2. And the trigger for the brain to do that is the presence of carbonic acid in our plasma. Acids, let me get back here. Acids are proton donors. They're proton donors. They release hydrogen ions. The more readily it gives up a hydrogen ion, the more acidic it is. See, when an acid dissociates in water, it will give, like hydrochloric acid gives up the hydrogen ion and the chlorine ion. The more hydrogen ions it gives up, the more acidic the acid is, the stronger the acid is. A very strong acid will give up lots of hydrogen ions. We call them protons. Because think about it, hydrogen, one proton and one electron. If it doesn't have its electron because it's now an ion, if it's a hydrogen ion, it doesn't have the electron. All that's left is the proton. So we call them protons. So we say acids are proton donors or hydrogen donors. I mean, think about it. Several treatments for acid reflux. Now, acid reflux is a condition that occurs when our stomach overproduces hydrochloric acid. Several medications for acid reflux are what are called proton pump inhibitors. They block the production of hydrogen ions. That means they're making less acid. So proton pump inhibitors reduce the amount of acid produced by the stomach by preventing the formation of protons. Hydrogen ion is nothing but a proton. So the key acid there is hydrochloric acid. So if acids give up protons, bases accept protons. Bases are hydrogen acceptors or proton acceptors. Anytime there's a hydrogen ion in solution, they will grab it. Sodium hydroxide will grab protons in solution. Sodium hydroxide is a base. If you mix a base and an acid together, you will have a neutralization reaction. The sodium hydroxide dissociates in water to give you the sodium ion and the OH or hydroxyl ion. If you mix an acid like hydrochloric acid together with the sodium hydroxide, you will get a reaction. You get a neutralization reaction. The sodium hydroxide dissociates into sodium and the hydroxyl unit. The hydrogen ion from the hydrochloric acid combines with the OH to form water, and the chlorine combines with sodium to form salt. We've neutralized the acid with the sodium hydroxide. In our bodies, there are two important bases we deal with. Just like the two important acids, carbonic acid and hydrochloric acid, the bicarbonate ion from sodium bicarbonate is a base, and ammonia is a base, because ammonia is what we get when we break down proteins, and the bicarbonate ion is what we get when we break down sodium bicarbonate. So in our bodies, the important bases are the bicarbonate ion and ammonia. The important acids are hydrochloric acid and carbonic acid. We measure the concentration. Now, this is something even more relevant now. We measure the concentration of acids and bases in our body using what we call the pH scale. And you probably, if you've had any kind of chemistry or science classes in the past, you probably heard of the pH scale. pH scale is a measure of the concentration of the hydrogen ions present. The bottom line is, the more hydrogen ions in a solution, the more acidic that solution is going to be. pH scale is a negative log scale, ranging from 0 to 14. Negative log means that as the hydrogen ion concentration in a solution increases, the pH drops, the number drops. So a pH of 0 or 1 is very acidic, because a pH of 1 means there's lots of hydrogen ions available. It is the inverse, the negative logarithm. Don't worry about this. Remember to say, well, of course, pH scale is the negative logarithm of the hydrogen ion concentration in moles per liter. I can't tell you how many times somebody's asked me that question, because they never have. So, but the point is, it's negative log scale. What does that really mean? It means that if the scale goes from 0 to 14, 7 is neutral. The hydrogen ion concentration at 7 is the same as the hydroxyl ion concentration. So it's neutral at 7. Distilled water is neutral. If you have a pH of 6, there are 10 times as many hydrogen ions at 6 as there were at 7. So it's a solution that has a pH of 6 is 10 times more acidic than 7. Something that has pH of 5 is 10 times more acidic than 6 and 100 times more acidic than 7. And if you go down to 4, something with a pH of 4 is 10 times more acidic than 5, 100 times more acidic than 6, and 1,000 times more acidic than 7. So it's a 10-fold change. As you go down the scale towards 0, you get more and more acidic. So something that's a pH of 1 is very acidic. Something that's a pH of 6 is mildly acidic. So it says like 5 is 10 times more than 6. So this is how so if we say that the pH is dropping, the acidity is getting greater. As we get closer and closer to 0, where we have more and more hydrogen ions available. So we measure anything above 7 is basic. The acidic side of the scale is 0 to 6.99. Anything 7.01 to 14 is basic. If you're basic, you have less and less hydrogen ions and more hydroxyl ions. And don't get hung up on that. As you know, if it gets closer to 14, it's a base. If it gets closer to 0, it's an acid. Neutral is 7. Our body ranges between 7.45 and 7.35. We are slightly alkaline. 7.4 is what our blood pH normally should be. As long as we stay within 7.45 and 7.35, we're going to be fine. If we get above 7.45 or below 7.35, we start getting in trouble. Our bodies don't like fluctuations in pH. And, while it doesn't sound like it's a whole lot, and it really isn't a whole lot to go from 7.4 to 7.35, or if we go below 7.35, that's a significant drop. Our bodies don't like that. It means that we're becoming more acidic. And, we're not talking a dramatic drop like from 7.4 down to 1. That'll never happen, at least you remain alive. But we get below 7, we go from 7.4 down to 7.35. We're becoming more acidic. Our bodies, have our tolerance to that is very, very low. We don't like that. We start getting below 7.35. If we move down lower, if we move below 7, we start having some real chemical problems in our bodies. The same if we go up the scale. If we get above 7.45, again, we start having some significant chemical issues. So we like to stay right around 7.4. We like to stay actually slightly alkaline. To give you an idea of what some things that we encounter on a daily basis, their pH, oven cleaner, 3.5 pH of 3.5, the top, near the top of the scale, right below sodium hydroxide. Ammonia, pH of about 11. Bleach, around 9.5. Egg whites, if you had eggs today for breakfast, the whites have a pH of 8. Milk has a pH of about 6. Coffee, pH of 5. Wine, pH of 3. Lemon juice, 2. Hydrochloric acid, stomach acid, 0 or 1. So these are all things that we encounter on a daily basis. If you like a soft drink, like Coke or Pepsi or Mountain Dew or something like that, pH of around 2 or 3. Pretty acidic stuff. So. I already said that. There we go. Okay, now, and anytime we combine an acid and a base, and we do this all the time in our bodies, we neutralize strong acids and strong bases so we can stay closer to our 7.4 pH range. And for example, if we want to neutralize a strong acid, we can use a base like sodium hydroxide, the example I gave. That will give you a salt and water. Neutralization reactions always end up with a salt and water. And we use these buffers. We call them buffers, and we use buffers all the time to neutralize our, when we get too acidic. But yes, we'll breathe faster when we exercise to get rid of CO2. But we've already got that carbon dioxide, that carbonic acid we form, so we have to neutralize that. And we usually neutralize that with bicarbonate. And we come back up to 7.4. And don't worry about that. We're going to, just remember that a buffer is something that will neutralize a sudden change in pH. So let's take a look at the organic side. The organic molecules are much more interesting than the inorganics. Organic molecules are all based in carbon. All organic molecules are based on carbon. The only molecules that have carbon that are not organic molecules are carbon dioxide and carbon monoxide. Carbon dioxide and carbon monoxide, they are not bound to other carbon atoms. They're only bound to oxygen. CO2 is bound to 2 oxygen molecules. Carbon monoxide, CO, is bound to 1 oxygen molecule. They are not organic molecules. They are the exception to the carbon rule. Everything else is bound to carbon. Carbon is the ideal atom for organic molecules. It shows up in all, it is the molecule of, it is the atom of life, if you will. When carbon combines with oxygen, with hydrogen and oxygen, you get the carbohydrates, the lipids, the proteins, and the nucleic acids. It shares its electrons. It doesn't kick out electrons. It doesn't grab electrons. It forms up to 4 covalent bonds with other atoms. And usually in this case, it's going to be carbons and hydrogens and oxygens. Now, the 4 organic compounds are the carbohydrates, the lipids, the proteins, and the nucleic acids. Organic compounds are long molecules. They can be hundreds of carbon atoms long. They can be made up of, they can have carbon and hydrogen and oxygen and nitrogen and phosphorus and sulfur all thrown together. They can be just carbon, hydrogen, and oxygen. They can have there's any combination here. The organic molecules are called polymers because they're so long, and they are made up of repeating units called monomers. They're linked together. So the first type, the carbohydrates, we look at all the starches, all the sugars, all the sucrose, the glucose, the milk sugar, the malt sugar, fructose, like high fructose corn syrups in our soft drinks, like in our Cokes or Pepsis or whatever, or Dr. Pepper or whatever your favorite is. Most of them are sweetened with high fructose corn syrup. All those sugars and all the starches in potatoes, and fruits, and vegetables, in flour and wheat, the starches that we consume in our breads and our pastas and our rolls, in our desserts, all these things, all these starches. So these are the carbohydrates. They consist of just long chains of carbons and hydrogens and oxygens. We have essentially a 1 to 2 to 1 ratio. One carbon, 2 hydrogens, and 1 oxygen in repeating sequence. What do we use the carbs for? It is the primary source of fuel for our bodies. The simple sugar glucose is what we use for our fuel. It's also structure. The DNA molecules, for example, are made of long chains of carbon atoms linked together. Proteins are long chains of carbon atoms linked together. Fats are long chains of carbon atoms linked together. Now, if you are a carbohydrate, you are in one of 3 categories. You are a monosaccharide, which is a simple sugar. You may have 4 carbons, 5 carbons, or 6 carbons. That's a simple sugar. A monosaccharide is the monomer of the carbohydrates. Remember, the polymers, long chains of carbons linked together. Well, the polymers are made up of monomers, smaller units that link together, link together, link together, link together. So the monosaccharide, a single sugar, glucose, is a monosaccharide. Glucose is a 6-carbon sugar. It is the sugar we use for fuel in our bodies. And so that is a monosaccharide. If we start linking glucose together, if we link 2 glucoses together, we get lactose, a double sugar. If we link glucose and fructose together, we get sucrose, table sugar. The monomers are the smallest type of carbohydrate. The monomer can be a 5-carbon sugar, like you see in DNA. It's called a ribose sugar. DNA is deoxyribose nucleic acid, or RNA, ribose nucleic acid, 5-carbon sugar. Disaccharides are 2 sugars linked together. Sucrose, table sugar, the stuff you get in the little white packets when you go to Weigel's or Pilots to get your coffee, and you want to put sugar in there, so you grab a couple of those things, little packets, tear them open, dump the sugar in. That is table sugar. It's sucrose. It's the stuff you go down to Food City and buy in the 5-pound bag. And whether it's Food City's brand or whether it's Domino's, it's the same stuff. It is the same stuff. The only difference would be that if the sugar says pure sugar or pure cane sugar, pure sugar usually comes from beets. Beet sugar, we can't tell the difference. If it says cane sugar, comes from the sugar cane plant. Pure sugar comes from, if it just says pure sugar, it comes from the sugar beet. Pure cane sugar comes from the sugar cane. You can't distinguish between the two. At least I can't. Most people can't. They look the same. They taste the same. A 5-pound bag of cane sugar and a 5-pound bag of pure sugar, same thing. So you can pick and choose whatever you want. You can get the yellow bag, Domino's bag, or you can get the blue bag from Food City or Walmart. Same sugar. Sugar is sugar. It's still sucrose. It's still 2 simple sugars linked together. Sucrose is made up of glucose and fructose linked together. Now the polysaccharides are lots, a technical term here, lots of sugars linked together. Lots of sugars linked together. Lots of simple sugars like glucose. The polysaccharides that we deal with in our bodies are usually starches and glycogen. Now, we eat the starches from plants. We store our polysaccharides in the form of glycogen. So there's a monosaccharide. There is a 6-carbon sugar. It's usually in some sort of ring structure. You have the carbon atoms are the black structures, the black atoms there. The red atoms are the oxygens, and the silver atoms are the hydrogens. It's C6H12O6. That is its basic formula. Structurally, there may be some variations on here, but it's going to be, most simple sugars are 5 or 6 carbon atoms. So these are the monomers. These are the building blocks of all the carbs. Our pentose sugars are 5-carbon, ribose and deoxyribose. The hexose sugars are the 6 carbons. Glucose, this is our blood sugar, blood glucose levels, blood sugar levels. This is what our bodies use for energy, as well as fructose and galactose. This is what the monosaccharides look like. Glucose, fructose, and galactose, they all have 6 carbons. They're all arranged slightly differently in their structure. Fructose has a carbon chain on each end of its 5-sided molecule. It still has 6 carbons. Here is glucose, which is a 6-sided molecule, and it still has a chain sticking off the side, a group sticking off with the 6th carbon. Galactose is very similar in structure as glucose. Just a few fundamental differences here between the two, just very few. So all these are 6-carbon sugars. Combine any one of these with another one, and you'll end up with one of the disaccharides. The disaccharides are the double sugars. They're big, they're bulky. See, now glucose works as our fuel because it's a small molecule. It can go through openings in the cell membrane. It's a large molecule. it has a molecular weight of 180 grams, or 180, because if you add up the atomic weights of all the carbons, hydrogens, and oxygen, it comes up to 180. It's a big molecule. You combine that with the second one, and now you're dealing with an even larger molecule. Sucrose is too big, or milk sugar, lactose is too big to get into the cell. And even glucose, the simple sugar, has to have a special passageway to get into the cell membrane. It has to have a carrier protein, which is an opening in the cell membrane, because the glucose is too big. It has to have a special channel to get in. It can only get in with help. That's the only way glucose can get in. How does the cell know to make these channels? Insulin gets released. When our blood sugar levels get too high, insulin gets released and tells the cell, make some channels to let the glucose come in. That's what it does. That's essentially the primary role of insulin. Sucrose can't get in a cell membrane. Sucrose is too big to fit through any of these proteins, these channels, these passageways that the cell may build. So we have to break it down to glucose. Sucrose, maltose, lactose are all double sugars. They're all, two simple sugars linked together. Again, table sugar, the stuff you buy at Food City or Walmart, the stuff you get in little white packets at Weigel's or at Pilot, that's sucrose. Glucose and fructose gives you sucrose. So you take the two 6-carbon sugars and you get the 12-carbon sucrose. Glucose and fructose give you sucrose, table sugar. Lactose is glucose and glucose. Lactose is galactose and glucose linked together. Milk sugar is formed from galactose and glucose. And maltose, malt sugar, used in baking, used in brewing alcohol, particularly beers. Maltose is simply two glucoses linked together. It doesn't matter what any of these are. The key ingredient here is glucose. Glucose shows up in all these. All these are going to get broken down to glucose so that we can use glucose in our cells, because glucose is the sugar molecule that our bodies depend on to get their energy. So glucose combined with fructose or itself or galactose will give you sucrose or lactose or maltose. Plain ordinary old table sugar is glucose and fructose. The third category of the carbohydrates are the polysaccharides. This is where we have the starches and the flours. This is the potato content. This is the corn. This is the cornmeal. This is the contents of what we eat when we eat potatoes or corn or fruits and vegetables. Root vegetables, potatoes and corn and potatoes and carrots and sweet potatoes and turnips and whatever else grows in the ground that we eat. All of these vegetables are going to contain polysaccharides. Cucumbers, if you enjoyed cucumbers and zucchini over the summer, or squash, if you had squash in your garden, or zucchini in your garden, or cucumbers in your garden, you were getting polysaccharides, getting the starchy portion of those vegetables. Polysaccharides are made up of monomers of the monosaccharides, usually glucose. Usually a whole bunch of glucoses linked together give you the polysaccharides. Polysaccharides are long chains of monomers. usually long chains of glucose. Starch. Starch is what a plant stores in its leaves, in its roots, in its fruit. Plants undergo photosynthesis. They produce glucose in photosynthesis. They store their glucose in the plants, in the leaf cells as starch. They store the glucose in the roots, in the tubers like potatoes and carrots and sweet potatoes as starch. They store it in the fruit, like in apples and in corn. Well, not corn, corn's a vegetable, but still, we store it in there. We store the starchy portion of, we store starch, plants store, let me try this again. The stored energy from the plant is contained in the starch. That's how they store it. Whether it's the pulp of an apple or the middle of the potato or the carrot or in the leaf itself, that's where the starch is contained. That's the stored energy for the plant. Photosynthesis involves using sunlight, the energy from sunlight, to combine with carbon dioxide and water and make glucose. That glucose gets stored as starch. We eat plants and we get that starch, whether it's wheat, when we have bread, rolls or bread or pasta, whether we're eating wheat flour or whole wheat flour, it doesn't matter. All the breads and pasta type foods that we eat are all contain carbohydrates that came from plants. Animals don't use starches. Humans don't use starches to store energy. We store our energy, our excess glucose, in the form of something called glycogen. Glycogen is a polysaccharide. And when we have excess glucose in our bodies, if we take in excess glucose, excess in the sense that the cells take all they want, and what's left over is then converted to glycogen and stored in the muscle cells and in the liver. So if we have extra glucose, we recognize that as, hey, that's a good source of energy. Let's save it. And we store it in the muscle cells as glycogen. We store it in the liver as glycogen. If we have even more glucose, we start storing it in the adipose tissue as fat. We convert it to fat. We also find that fiber is a type of polysaccharide. Fiber in foods is made from cellulose. Cellulose is a type of polysaccharide. It's what makes up the cell wall of plants. We can't process cellulose. It's indigestible. We call it the fiber. We call it fiber. We eat high fiber foods. We eat a salad. If you like spinach or romaine lettuce or any kind of salad, doesn't matter, any kind of food like that, high in fiber. If you like whole wheat bread is called whole wheat because when wheat is grown, it forms a kernel, sort of like corn forms a kernel, a different type of structure. Forms a kernel with an outer covering. The husk around the wheat grain is called the bran. Now, if we have 100% whole wheat bread, we have included the bran into the flour. And so that whole wheat bread is higher in fiber. We can't process the bran, it just passes through us. Most breads, well, I should say, white bread has the bran removed. The husk covering of the wheat kernel is removed because it's cellulose. We can't process that cellulose. It's a polysaccharide. We can't process it. The husk, the wheat bran will pass through us. Consider this. If you like corn, if you like corn on the cob, for example, corn on the cob, the corn kernel has an outer covering. The yellow portion or the white portion of the kernel is made of cellulose. It's not digestible. You bite into it, you eat it, you get the contents of the kernel, you eat that, and it contains the starch and water, and it's very tasty and you enjoy it. Well, what will happen is the next day, your fecal matter will contain the undigested kernel, the covering of the corn kernel. You can see if you're inclined to observe, you can see the outer covering of the kernel, the yellow portion of the kernel in your fecal matter, while the contents, the starchy contents, have been consumed or used. And so we don't process the outer covering. It is undigestible. It is still a high fiber food. So anyway, this is what they look like. For example, starch and glycogen look very similar. They're branched, long chain polysaccharides with each one of those little six-sided, that little hexagon structure there, all linked together. Those are all glucose molecules. And so we have lots and lots and lots of glucose molecules. However, when glucose is linked together in straight chains, like it is at the end, that's cellulose. For whatever reason, we can't break that apart like we can break apart starch or break apart glycogen. I mean, our bodies can take glycogen and rupture it and convert it to glucose. Insulin released from our pancreas tells the cells to open up and let glucose come in to the cell and lowers our blood glucose. But what if our blood sugar is too low to begin with? Then we can release another hormone from our pancreas called glucagon. Glucagon tells the liver to break down glycogen into glucose. And so we release glucose. The same glucose in glycogen is found in starch. Same glucose in starch is found in glycogen. Glucose is glucose. We can break down, we can eat starch. We eat pasta and breads and grains all the time. We can break that starch down to glucose. And that's great. We can't store excess glucose as starch, but we can store it as glycogen. But when glucose is lined up like it is in cellulose, we can't eat it. We can't go eat grass and process it. We can't process the cellulose portion of grass. The cellulose portion of any fruit or vegetable doesn't get processed. It becomes the fiber. And the difference between cellulose and lignin, which is wood, is, lignin is mixed in with cellulose in wood. We can burn wood. I mean, look at all that. Look at that cellulose there. Every one of those glucose molecules represents a lot of stored energy. What do we do with wood? We burn wood, or we can use it for structures. But if we burn wood in our fire pit or our fireplace, it releases energy, and we like that. We just can't get the energy to eat with it. So, okay, I want to make a couple points here on lipids, and then we'll call it a week as far as lecture goes. Lipids, the fats. Carbon, hydrogen, and oxygen. Not quite the same ratio that we had with 1 to 2 to 1 in carbohydrates. Sometimes we'll have phosphorus in there to alter the state of the fat. Sometimes we're going to have, it'll be covalently bonded, but we have double bonds in it instead of single bonds. Carbohydrates are bound by nothing but covalent single bonds, sharing electrons covalently sharing 2 electrons at a time. Sometimes in fats, we share more than 2. Sometimes we share 4 electrons. So something else about fats is that they are not soluble in water. If you mix up oil and vinegar, making a simple salad dressing, oil and vinegar, the oil will float on top of the vinegar because the vinegar is mostly water and a little acetic acid. The oil floats there because it is not water soluble. Things that, water cannot go into oil, oil cannot go into water. They are insoluble to each other. So the oil floats on top of the water. That's what it does. Just sits there and floats. You can shake up the oil and vinegar and scatter the oil all through the oil and vinegar mixture, and then the oil is all going to migrate to the surface of the mixture, of the solution. Not going to go in, into the vinegar. 4 categories of lipids. The triglycerides, term you may have heard, is what we generally call the fats or neutral fats. If we say fats, we're usually talking about triglycerides. So if I say fat instead of lipid, I mean the same thing. Phospholipids, the stuff that makes up our cell membranes. Our cell membranes are what we call a phospholipid bilayer. We add a phosphorus atom to a fat, and it forms this layer, this double layer, what makes up our cell membrane, phospholipids. Steroids are the 3rd category. Steroids are important chemicals in our bodies make up important chemicals called hormones. They're also used for structure. The most important steroid is cholesterol. Cholesterol is not evil. No food is evil. Sugars and carbohydrates are not evil. Cholesterol is not an evil compound. All has to do with moderation. We'll talk more about that next week. Steroids make up the sex hormones, estrogen and testosterone. Steroids make up a number of hormones released from the adrenal glands that control water levels and blood pressure and sugar levels and help trigger the onset of puberty. The steroids then are a type of fat, very specialized type of lipid. It has a 4 or 5 ring-shaped structure. All steroids look very, very similar. They're all derived from cholesterol. The difference between testosterone and estrogen are just some minor changes in the chemical groups attached to the ends. The last category are the eicosanoids. The eicosanoids are a type of lipid often used as an anti-inflammatory compound. So when we deal with inflammation. So last point, the lipids. As we lipids here, we take a look at the triglycerides, the neutral fats. Terminology here is pretty specific. If the lipid is a solid at room temperature, it's a fat. If it's liquid at room temperature, it's an oil. And both types of the triglycerides are made up of fatty acid chains attached to a molecule called glycerol. And I'm going to stop because it's almost 12:30 and did pretty good today. So let's see. Any questions on this? We'll get some good examples of fats and stuff like that next week. So we'll pick up with this on Tuesday. We may wrap everything up on Tuesday. We'll definitely wrap up the chapter by Thursday. Lab this afternoon is going to be on the skull. And I'll talk a little bit about what your expectations are for the bone test, which will come up next month. So, and the lab test is open for open today for the Thursday lab, and it's open for 10 days. So, any questions on anything? If not, I want to get us out of here. I'll see some of you in lab today, and otherwise have a good weekend.